Welcome to yet another journal club brought to you by Lifespan.io. Dr. Oliver Medvedik, and as per usual, we'll be presenting a journal article addressing something a little bit more interesting, well, a little different than usual. We're looking at a different compound that we haven't looked at before, which is CBDs and THCs, so tetrahydrocannabinol and cannabinoids in general. As we're all aware, these are compounds found in cannabis sativa, cannabis indica, and the various cannabis strains, and lots of products have CBDs in them. And this paper is basically looking at phytocannabinoids, these classes of compounds, and their effects on senescence. I'll mention, though, that having read this paper, there's any questions or comments, please use the raise hand icon at the bottom. There's a lot to be desired as far as figuring out exactly what the effects are on senescence in this paper. So there's quite a bit of data, but there's a lot of questions here. And I think for me, I think this is an interesting area of research because not much is really being, I think, done so far with these compounds with regards to cellular senescence and aging. These compounds are found in a lot of products, a lot of creams, a lot of topical ointments. So figuring out what these compounds are actually doing is important, I think. So that being said, let me just share my screen here. So this is the paper. Phytocannabinoids stimulate rejuvenation and prevent cellular senescence in human dermal fibroblasts. And I'm not really sure, having read the paper, if they actually prevent cellular senescence in human dermal fibroblasts. They do have some interesting effects, but I think this paper raises a lot more questions than I expected. The human dermal fibroblasts, I believe these are, the authors use these CCD1064SK skin fibroblasts, which I believe are human foreskin fibroblasts. And they have this model here, this SIPS model, stress-induced premature senescent model, where they hydrogen peroxide treat these skin fibroblasts to induce a senescence-like state. And then they test a variety of these phytocannabinoids, PCBs, on these fibroblasts, and see if there's any improvements in their in their morphology, in their behavioral, so on and so forth. So by the way, anybody on this panel read this paper other than myself? Have a chance to look at it? Nobody.
I didn't myself, no.
Okay.
I only skimmed it, but very briefly.
Okay. Yeah. So, I'll just basically jump to the data. So they look at a number of different things using these compounds. They actually also compare to a variety of compounds that we've already heard about: metformin, rapamycin. That's actually, for some reason, it's in the supplemental, even though it's kind of an important control, right? So first off, there's a lot of kind of interesting effects here, or odd effects, that it's kind of hard to account. So one thing that needs to be mentioned is that these cannabinoids are resuspended in a carrier, DMSO, dimethyl sulfoxide. A lot of you have heard of this compound. It's used as a cryoprotectant. It's actually used in a lot of as a carrier in a lot of medications, right? So it's basically it's allows things to essentially pass through the epidermis and get into pass through cellular membranes. And in some of the figures in this paper, it seems to have an effect on its own, which is, I'm not sure if you would call that worrisome or interesting, but I think that needs to be kind of addressed because it's used in a lot of topical treatments, and yet there's an effect. So the first thing that they do is they do a beta-galactosidase activity to see essentially. So they look at, let's see, on these. So they look at human dermal fibroblasts after population doubling, 48 doubling. So these are in a normal, I guess, kind of senescent state after they've gone through population doubling 48. And they use this other version, and I'm not sure why they switch between the two, but they go through these fibroblasts that are, after 48 doubling, so they're senescent, and they have this hydrogen peroxide induced kind of premature state. And they test various concentrations of THC and CBD, so going from 0.25 micromolar to 10 micromolar. And I'm not really sure what that corresponds to as far as what you would normally see, in a topical ointment, for example. So this is after 2 days of treatment and 15 days of treatment. And what they note here is that for some reason, they look at several concentrations. There's this kind of odd bimodal effect with THC, and I'm not really sure what to make of this. You have increased beta-galactosidase activity, which is indicative of kind of increased cellular senescence. And this is happening with 0.25 micromolar and 5 micromolar THC. And the authors, let's see, and that's also happening with CBD. And I'm not sure, or at a higher concentration, and I'm not sure why there's this bimodal effect happening here with a high and low concentration. The authors kind of choose to ignore that and say that, since it seems to be inducing stress at those concentrations, they're going to stick with, I think, this midpoint concentration that they test later, which is 2 micromolar. So they're kind of ignoring this low and high concentration. Yeah, Warren?
I think they did mention about the high concentration, that in the end it's just toxic. I guess everything you just throw in a cell with a very, very high concentration would be toxic in the end.
Yeah.
They actually say that the last concentrations, it's lower beta gal because the cells are just doing nothing. They're just dying and not doing beta gal.
Yeah, so they mentioned that with the high concentration, but what about the 0.25 micromolar? Why is this bimodal? Why do you have this?
But maybe then, if you just consider that too much of anything is toxic, then you maybe can think of it as only one. So you have only one peak, which is real. The high peak is just throw something with too high concentration, so it's not really physiological.
Right. So they do mention that, beyond 5 micromolar or 7.5 micromolar, you start getting a toxicity effect, and you're right about that, Warren. That if you start overloading cells with too much of anything, it starts to get toxic to them. But you do have this kind of interesting at least for the beta-galactosidase levels here, at like a lower concentration, right? And I'm not sure what accounts for that. So let's say if we do ignore the high peak and say, well, maybe it's not a real bimodal kind of distribution. Maybe the high peak is just way stressed out. It could be anything. So you do, so I'm not sure why you would get this, this effect kind of at 0.25 micromolar THC, higher beta gal activity, but at the higher concentrations, 0.5, 1, 2.0, let's ignore 5.0 for both THC and CBD, you don't. And by the way, THC and CBD hit different receptors. So there are these cannabinoid receptors, I believe CB1 and CB2, and I think all of these receptors are G protein coupled receptors. Yeah, Mika has a question. Hand up.
If it was just toxicity due to sheer volume, wouldn't you expect to see the even higher volumes be also toxic? So you wouldn't see 5 but not 7 and 10. You'd see 5, 7, and 10 would all be toxic if it was just due to overloading the cells.
Yeah. Well, at least for the beta-galactosidase results here, yeah. So it does go down, you're right, with 7.5 and 10. But there's, let's see, Oren, you had a question. Let me take a, oops. Yes, Oren.
But with regard to what Mika said, that's what they explained, that they say that it's not really going down the beta-gal, it's just the cells are just totally, I don't know, not doing what they're supposed to do, and they also don't have the beta-gal activity, so they're just completely ruined, and that's why you have lower beta-gal in the very high concentrations. That's, I think, was their explanations for why it's going down in the end, and it's like you shouldn't think about it as healthy cells. You should think about it as completely dead.
Yeah, because that's in supplemental. So yeah, it says while 10 micromolar of CBD did decrease beta-gal activity, which might be seen as beneficial, this is likely due to decreased cell number by the severe cytotoxic effect seen via phase contrast microscopy at higher concentration. So my kind of I'm gonna stop sharing here for a little while, but I would say that there are methodology and presentation issues I found with a lot of kind of the figures here, and I think that's one of them, because that's in the figure here where it says because of higher cytotoxicity, which addresses what you said, Oren, and they mentioned that in the paper. And then I'm looking at the beta-gal levels, and you can see the levels go down. I think to properly show that data, you have to normalize it, right? Otherwise, you're showing those two figures, right? And you have the supplemental, which is separate, which shows increased cytotoxicity. But I think that data should be combined, right? It should be normalized. So those histograms actually look the way they're supposed to look, which is the beta-gal activity levels, the beta-gal levels actually are probably higher, right? They're, and that should be I guess people agree or disagree with that. I think the data should be presented in or should have been presented in that way to make it a little bit more clearer, because they do mention it, but I would normalize it just so you don't have to have it separately mentioned in the text and have it in supplemental and then have to refer back to that data. Just combine the two, and that should be possible.
Is the idea here that they can look at the number of cells that are actually alive and functional and then normalize on that? Is that what you're suggesting? Like normalize on the number of functional cells rather than the total population?
Yeah. I mean, because it's yeah. I mean, does that make sense?
I think it's reasonable. I don't know enough to know if that would be on the flip side. If I was going to play devil's advocate against that, I would say you want to be careful that you're not making too many assumptions, and we think these cells are dead, but we're not really quite sure if they're dead. We think they're non-functional, we're not quite sure. I don't know how much confidence they have in that second number that you're trying to normalize by, and maybe they just weren't confident in it, and so they're worried about normalizing against something they don't have any confidence in.
Well, you should be able to do a viability assay. That's kind of standard, right, for a lot of cell cell biology research. There's a number of different dyes that could be used to basically measure whether or not cells are taking them up, and if they're not taking them up, then they're not viable. Gotcha. Yeah.
So if we assume that the drop off at the end was due to toxicity, should we be concerned that the effective dose was so close to the level of toxicity? Like, the fact that you don't see any real benefit right until 7.5 and then or until 5 and then right at 7.5 the cells all die. It seems dangerously tight, if I'm understanding this correctly.
Yeah, well, I'm not sure if it's in this case dangerously tight, although that tends to be an issue with a lot of other drugs, especially I would say especially with cellular reprogramming factors. I'm not sure. Again, I'm not sure what the normal dosage is for THC or CBD when it comes to skin samples. But be that as it may, I think they choose to ignore those values, those higher levels, and for their subsequent studies, they go on with sort of a middle dose that's someplace kind of within this valley, this histogram valley, if you will. Okay, so kind of the most potent effect that they see is they basically do they look at cellular viability in these CIPS fibroblasts, which are these senescence-induced fibroblasts. Let's take a look at here. So they do two. Actually, there's two kind of two important. Well, there's a couple of important figures, but figure 2 and let's see, where's the figure 4 is the wound healing test. So they do this this MTT assay, which basically tests for essentially, see, it's an enzyme that undergoes reduction. So you could use this to measure cellular viability, which I guess they could have done it with the other cells, right? They could have used that as a cellular viability assay. So they're looking at healthy cells, which I believe are the same cells, but population doubling 24. So non-senescent CDCD 10-0 or 1064 SK human dermal fibroblasts, and the ones that they call CIPS, which have been treated with hydrogen peroxide. And they use these hydrogen peroxide treated cells as sort of, I guess you could say, an accelerated senescence model. I'm not sure why they don't use like high population doubling cells here, but they use these. They use the ones that are treated with H2O2, these CIP cells, and they do a viability assay, this MTT viability assay, and they compare healthy and senescent cells. And kind of the interesting thing for me here is I'm not sure. So they look at healthy cells, and I think they state that there's a significant difference, albeit very small, between DMSO controls and THC at 2 micromolar concentrations for the healthy cells, and this is CBD, and I'm not sure if there's. I think they mentioned there's not much of a difference for CBD. There is a much more pronounced difference with senescent cells, with THC at 0.5 micromolar, even more at THC 2 micromolar. So one question, so it seems to be not having or having a negligible effect on healthy cells, but it seems to be these compounds, at least at this concentration, THC and even CBD in this case, on these stressed or these hydrogen peroxide treated fibroblasts. I don't know. My question is, to folks out there, is that a good thing if you want to be promoting the viability of senescent cells? Because we've talked before, obviously, about senolytics and using agents to, I'm going to stop sharing here, agents to clear out senescent cells, but here we're making these cells more viable, right? So is there kind of a bit of a conflict here for what we expect THC or CBD to want to do with senescent cells?
Do they have any data on whether the cells are not just viable, but doing the things they're supposed to be doing otherwise, or are they just like, hey, they're not dead?
They do that later. They do a wound healing assay, and I believe that Hey, John. So happy New Year. He's signing off right now, one of our members. So yeah, I think they have an improved wound healing capability. So their their functionality in that respect is improved. I think as far as functionality is concerned, they look at a whole bunch of other markers, but that's the only other kind of cell-based functionality test is done.
For me, that's the thing that For me, that's the thing that matters: is the cell behaving like the cell that it's supposed to be, or is it behaving like a senescent cell? Like, is it secreting SASP and all the other bad things?
Yeah, they don't.
Or is it doing both?
Yeah, that's one thing. That's one thing that I wish was done more later in the paper. They look at the expression of a variety of factors, but I kind of wish that they looked at a whole bunch of more factors, like a lot more senescence associated secretory phenotype factors. I think this paper kind of opens up a lot of questions as to better characterization of these cells. And we're also looking at one cell type. We're looking at these foreskin fibroblasts, right? So I'd like to see a whole bunch of and I think the reason that they choose these cells is that they're interested from a dermatological perspective, a cosmetological perspective, and I think these are used probably in those types of assays because they are skin fibroblasts. But it would be interesting, obviously, to see senescent cells from a variety of tissue types and see, how these effects translate throughout different cell types. So, so there are limitations to this. We are looking at one cell type, and we're looking at this one senescent cell. So one cell type, and the senescent state is induced by this hydrogen peroxide treatment, right? Or in the other case, population doublings after I think 48 doublings, which would be a kind of a more natural senescent state. So I'm not sure why that wasn't kind of looked at, and why this H2O2 system was kind of focused on instead.
Yeah, I tend to agree with your assessment there. I think that I would have much rather seen a natural age state. It's possible that the type of the state the cell was in was recovered, but a naturally senescent cell would not have been recovered, because there's many ways to get to senescence, and they don't all lead to the same thing. And so, unless maybe this is a great treatment for hydrogen peroxide burning. But,
yeah. So there's a lot left to be desired. So that's the data here. So so as soon as I saw that, I was thinking, okay, so at least for these H2O2 treated cells, it seems to improve their viability. But is that really a good thing, right? At least for senescent cells. And now they're looking at these crystal violet stained fibroblasts. So they basically are, let's see, CB stained fibroblasts. Are they? Yeah. So we're looking at the same cells here. Just double checking here.
You're not sure anymore.
They are. Yeah, I think it's the same cell type. Yeah. So they're looking at crystal violet staining. So this basically stains cells, and you can measure how well they're attached to a substrate. So healthy cells are basically stuck to the bottom of the surface plate, and unhealthy cells kind of detach, right? So so it's another measure of viability. So
did you mean to be sharing your screen? Or not sharing your screen anymore?
Oh, sorry about that. Yeah, if I have any more glitches like that, let me know. So let me share my screen here. I just scrolled down, so I didn't get to the data yet. There we go. So the crystal violet staining. So this histogram is basically taken from the top panel here, 24 hours and 48 hours after treatment with CBD and THC. So they stick with this 2 micromolar amount. So healthy cells and senescent cells. And what do they see? So if I'm reading this correctly, it does, So just like with the viability assay before, you do tend to see, let's see, so this is basically the crystal violet absorbance. So there's increased viability in senescent cells, and they're also looking at beta gal activity here. Let's see, is this a drop compared? I mean, it is a drop. I'm looking at here, H2O2 and THC in the senescent cells. But do we see the opposite before? So interesting that the beta gal activity goes down. They say that the beta galactosidase activity goes up slightly. I don't know how real that is in healthy cells, which is maybe a little bit worrisome. There doesn't appear to be effect in improvement in adherence to the bottom with healthy cells, but there does with the senescent cells. Let me take a look at because it says here that the beta gal activity for senescent cells drops. And if we go to the first figure, but these are normal cells. Okay. Well, yeah, it's kind of comparing two different things. So these are population doubling 48. So these are like normal. Yeah, these are sort of like normal cells that have been kind of aged by going through multiple population doublings, and these are the H2O2 treated cells. So I'm not really sure like what the kind of the fundamental difference is, between kind of all of the pathways for these CYP cells that they refer to that are treated with H2O2, and kind of cells that have undergone cellular senescence through multiple rounds of population doublings. I think the take-home message from this is that the viability also, by using this kind of adherence assay test, the senescent cells are kind of their viability is improved. Now, whether or not, interestingly, if the beta galactosidase activity goes down in these senescent cells, is there also a concomitant decrease in a whole bunch of other senescence factors? And they look at some later on, like P21, for example, P16, but kind of not the full gamut, right? So like a whole panel where you would normally look at hundreds of different, like an expression profile. So it would be interesting to see if these senescent cells are being reverted to more of a non-senescent state, and I'm not sure about that. So that's kind of led me to scratch my head and say, well, do we want these senescent cells to be hanging around if they're truly senescent, or are they actually being reverted, right, to something a little bit less senescent, right, and not being problematic to surrounding cells. So I'm not sure about that. Next, they do a wound healing assay, and this is kind of where you see the most pronounced effects. So this assay is basically, you grow cells on a plate and use some sort of a tool, like a pipette tip or something, to basically make a scratch across the surface. So you basically abrade the cells. And the cells, as they replicate, they fill in that gap. So you can basically measure the percentage of that gap being filled. And then the cells stop dividing. So they normally have contact inhibition. So they keep dividing until they sort of tile in that area. And that's kind of a basic, what they refer to as a wound healing assay. And that's been done for a long time, I believe. And that's sort of a typical assay for that. So they look at healthy and senescent cells. And again, the senescent cells here that they're referring to are these H2O2 treated cells. Now, one thing that keep in mind is when I looked at these assays here, I looked at percentage of unhealed wounds. So percentage is basically how much of that space is kind of filled in. And then when I looked at these senescent cells, it went past 100% to like 200%. And I was like, how can it be 200% unhealed? This means like even worse. It turns out that the authors explain it that for the senescent cells, I guess they have when you do this scratch, the ones on the edge tend to lift off, so you get more of a more damage to kind of more of a gap than you would that goes beyond the initial basically scratch. So that's where you get this higher percentage of unhealed wounds. So if the pipette tip scraped half a millimeter across, then that would spread and cells would slough off on the edges, and you would get more of maybe a 2 millimeter gap. So that's how the authors explain that. So this is post-treatment with these compounds, THC, CBD. I believe they use 2 micromolar. I think, 2 micromolar. That's the amount that they basically stuck with. And they do healthy and senescent, and percent of unhealed wound. So basically you want to see an improvement. So going down 0 is the cells are filling in. So it's kind of looking at it backwards. So percent of unhealed wound. And you sort of get this, you get a dramatic improvement with wound healing for healthy cells. And also you get this improvement, let's see, percent unhealed. Let's see, do you get a percentage unhealed? Let's see, yeah. Eventually, over a little bit longer time, you get an improvement in wound healing with THC and CBD. Interestingly, the DMSO seems to be having some sort of kind of a negative effect on wound healing. I'm not sure why. Dimethyl sulfoxide is used in a lot of topical ointments I think it's used in arthritis medications and a lot of other things. It's basically considered to be an inert carrier, but it would be interesting to see what other kind of effects DMSO might have, because it's clearly having an effect on its own. But be that as it may, it looks like CBD and THC, at least in this model here, are improving the wound healing in this assay. So again, this is a tissue culture based assay. It's not an animal, so take that how you want. Yeah, Mika?
I just wanted to mention what John said in chat before he left, that he linked to a paper that suggests DMSO actually is not inert at all. It has very significant changes or impacts.
Yeah. So I would add to that comment and say, like to see kind of more focus maybe on DMSO, itself, to see what effects it's having, because it is used in a lot of applications, and or maybe use a different carrier, right? If you're going to look at THC and CBD, and maybe if you want to ignore the DMSO effects, I'm not sure if you can use ethanol, or some other solvent, that might not have the effects that you're seeing, right? That basically might obfuscate some of these, some of the effects that THC and CBD are having. So anyway, there's improved wound healing. So I think that's kind of like that, and increased cell viability is like the more kind of pronounced effect. So are these senescent cells acting more normally than usual? Well, based on wound healing, perhaps. Okay. So here, here's a lot of data where they basically, it's related to the toxicity, and I'm not really sure how to interpret this data, really. There's a lot of, so they're basically looking at nuclear morphology, and the microscopic data, I believe, is in a supplemental, and I'm not really too familiar with this as kind of a parameter for healthy cells. Like, I do know if you have grossly aberrant nuclear morphology, that's a problem. So obviously, cells that have lamin A deficiencies and things like that, right? And but they're looking at nuclear area, and for healthy and senescent cells. And I'm not sure, looking at sort of, when I looked at this histogram, and we're looking at, claiming that there's a significance, maybe there's a statistical significance between the effects on kind of the nuclear, eccentricity, which I'm not sure what that's a measure of. I mean, I guess nucleus is more oval than circular, right? The nuclear area, obviously, how big the nucleus is. It's clear when you look at, H2O2 and DMSO, right? DMSO is having kind of more of a pronounced effect than the THC or CBD. So you're getting back to the DMSO, what we've been, what John had mentioned, that it has effects. It does seem to be having an effect, at least at the concentrations they're using it as a carrier. If we look at figure day 1A, untreated to DMSO, right? And then you want to compare CBD to DMSO, I think my major problem here is that you've got so much of an effect happening from the DMSO. I'm not sure if you can. I would, even if there is a significant difference between DMSO and THC, I'm not sure if I would trust that difference, just because the DMSO is having an even more pronounced effect if you compare to untreated, right? Than within the experiments. I don't know if anybody else wants to chime in with that, about that. Yeah, Mika.
Just a thumbs up. I agree. It's questionable. Like,
yeah.
If DMSO makes things worse than THC, brings it back to untreated.
Yeah.
It's hard to say if maybe the THC was just counteracting the DMSO, but didn't actually do anything itself.
Yeah. So if the whole focus here is to look at, strictly look at the effects of these cannabinoids, I wouldn't really delve too deeply into the woods kind of this figure, just because the differences do appear to be slight at best, and the biggest differences are with the control carrier, which is DMSO. So it's kind of really hard for me to make out exactly what's happening here, other than DMSO is having an effect. So again, back to the carrier. So here, yeah.
I just want to call out that I'm glad that the authors included the DMSO control, and this is a good example of why it's very important to do that. Like, even if they just left it out or didn't include it, we would have seen effects that could have potentially been misinterpreted. So I'm glad they did that.
Yeah, so props to the authors for that. I do have issues with with the significant differences, but one thing I must say for the authors here is that whether the data was good or whether the data was ugly, the authors presented it. So, and the fact that we got to see the DMSO data makes you realize that, hey, this type of control is very necessary. And I mention that to my students too when I run labs. It sometimes seems you can get lazy and not include DMSO control, right? So a lot of times people do experiments and they say, well, I've got untreated, and then I've got my experiment, which is, what I'm testing in the carrier, but without having the carrier as the control. And, a lot of times people don't include it just because they forget, or it's more of a lot of times it's kind of unintentional, right? A lot of times it's, sometimes a researcher or a grad student just doesn't realize the importance of having that sort of control, and you see this pronounced effect, and you're like, wow, this cannabinoid is having a really robust response. And here we see that a lot of this response is coming from DMSO. Why? I'm not sure, but it's clearly having an effect. So so I think an interesting experiment to do, if a very simple experiment. So a lot of times when we read these papers, authors do a lot of these background experiments, and sometimes they seem so repetitious and boring, like a whole figure where you just test 5 or 10 different carriers, right? And to demonstrate that they don't have any toxicity or any other types of issues among themselves before they actually go into the experiments. And you're like, gee, that seems so kind of redundant. Don't we already know which carrier to use, and can't we just jump into the experiments? But this kind of illustrates that that research kind of is a plodding, meticulous process, and a lot of people don't have the patience, right, to basically set up everything they need to set up before they actually go on with the experiments, and you could easily be led astray by not having the right control, and sometimes the right control isn't very obvious. So
Yeah.
So yeah, thanks for pointing that out, Mika. It's an important point, right? Okay, so what are they looking at here? I believe they're looking at.
You're not sharing again.
Sorry about that. So what we're looking at here is, I believe, expression levels of various proteins. So mRNA levels, and they get slightly different results so they do Western blots and they look at protein levels, and they get slightly different. They get different results. In some cases, the mRNA levels go up, and other times the corresponding proteins don't. And the authors mentioned that it might have something to do with the time at which they looked at the cells, which maybe they could do that a bit better next time, because there is always going to be a lag if you do going from your messenger RNA to your protein, right? It takes a bit longer for translation to happen. So they're looking at the expression of a variety of factors. So COL1A and COL3A1, I believe, encode various types of collagens. So these are fibroblasts, so they secrete things into the extracellular matrix, right? They make the extracellular matrix. ELN is, I believe, elastin, so another member of the extracellular matrix. MMP1, MMP2, I think, are matrix metalloproteinases, which are involved in cell motility. So basically, that's like a turnover of the matrix. HAS1, what's HAS1? Keep forgetting. I mentioned HAS1. It's probably something else related to the ECM.
Hyaluron.
I can never pronounce hyaluronic acid. Hyaluron synthase. So it's the other component of extracellular matrix. I think most famously, long chain hyaluronic acids are secreted by naked mole rats, which basically is kind of purported to give their kind of anti-cancer effects, is these long chain hyaluronic acids. So they're important molecules in the ECM. They basically are very hygroscopic. They retain a lot of water. So also involved in kind of capturing signaling molecules. And TIMP1, tissue inhibitor of metalloproteinases. So let's see what happens. So, well, if you treat them with DMSO, you also get an increase, a slight increase of collagens, but an even more so increase, so much more significant for THC. And I believe, I don't know, they don't, is it significant? I mean, it does look like it's hard to say if that's insignificant with CBD. So you do get an increase with this form of collagen. I don't know the exact difference between COL1A1 and COL3 and COL1. So different forms of collagen. There's a lot of different types of collagen. I think this might be the most common one secreted by fibroblasts. So
that went down with the senescence cells and up with the healthy cells. Is that expected, or is that surprising?
I don't think it was expected by the cells, because it seems that we had increased viability and increased in the previous assay of these senescent cells sticking to the dish. So I would expect that their ECM would be more normal, or basically their production of these factors would be, at least from the mRNA level, would be kind of better, meaning that you have more of an increase. So let's see, elastin goes up. Wow. So DMSO has a dramatic effect for some reason, on elastin expression levels in senescent cells. I'm not sure why.
Do we know, just back on the, sorry, I'm still on the collagen one, collagen 1A. So senescent cells produce more, am I reading this correctly, that they produce more collagen? Is that normal and expected? I would have expected less collagen when senescent, not more.
Are we talking?
Am I wrong there? Like just the untreated and the H2O2, no DMSO, no THC, no TBD. Comparing healthy versus senescent.
Well, I don't know what the total amount is, because this is a full change. So if you look at the y-axis, this for the healthy, it goes from 0 to 5, and for senescent it's much lower. It goes 0 to 2.0.
Yeah, but we're just looking at 1 versus 1.5. So that suggests to me that the collagen, I guess, what is this a full change from? What is the starting point? The starting point always the healthy untreated? Is that what this is a full change from?
It's a good question.
It looks like healthy untreated looks like it's 1 on all of them. Is that correct? No.
I think it's a housekeeping gene, GAPDH. So they do relative to this housekeeping gene. This is what the caption says.
Okay.
Yeah, I'm just trying to think here if that's the proper way to do it.
I just my instinct was that collagen production would go down when the cell becomes senescent, not up. So I was a little surprised to see that bar be higher. But again, I might be totally misinterpreting this chart.
Yeah, yeah. I, off the top of my head, I would say that that they would be producing. Yeah, they especially fibroblasts. They should be producing less, like kind of. Then again, these are H2O2 treated senescence cells that have induced senescence. So I'm not sure. Yeah. I'm not sure if population doubling at 50 or 48 would do the same thing.
Sure.
Yeah. So good. All good questions. Not sure. So it's a bit confusing. So again, you've got those DMSO effects, and I'm not sure exactly how much of that is kind of obfuscating some of the effects here, especially with I mean, it's a big difference here with elastin. So the matrix metalloproteinases, those let's see, what was HAS1 again? Oh yeah, hyaluronic acid. Yeah. I mean, there is an increase in healthy cells, not much of an increase with senescent cells, but the messenger RNA levels do go up. Let's see, do they go? No, they go down actually with senescent cells. So there are like these opposite effects. So you have this increase in matrix metalloproteinases, which I'm not sure how to interpret that. I mean, it's a good thing, right? Because this is used for rearrangement of the, if you have healthy cells giving off more, I guess potentially producing more collagen, more hyaluronic acid, you should also expect a concomitant turnover. So these enzymes have to go up. For the senescent cells in these assays, these enzymes go down, I believe. So, I don't know. Yeah, I'm not sure why you're getting that effect, right? So why you would get higher levels of, actually the levels really go up. It's the DMSO. It's really hard to interpret for the senescent cells.
Is it possible that, so because we're not doing the 48 doublings, we're doing the hydrogen peroxide.
Yeah.
Is it possible that maybe before the cells become senescent due to exposure to hydrogen peroxide, they react to the presence of the hydrogen peroxide and start changing their internal production, like as a defense mechanism perhaps, or as a consequence. And so what we're seeing is actually kind of leftover from that initial reaction to the hydrogen peroxide rather than the kind of long-term stable state. Like, because the mRNA doesn't disappear right away, right? Like,
yeah.
If it's produced, it's going to stick around for a while. So if they overproduced when that first, hydrogen peroxide molecule hit, and then they started producing more of these things, but then the cell eventually became senescent and then started producing less. But when we test it, you may see, it's got a lot of this mRNA in there because it produced it earlier. Is that a possibility?
It's possible. I mean, a lot of mRNA has a short, kind of fast turnover, but it could, the gene could be turned on for a while, so due to this treatment. So, I don't know how characterized this process is when they're doing these so-called CYPs treated cells, when they do this hydrogen peroxide treatment. Do they do like a kind of an analysis of the cells, like you said, like you treat them with H2O2, you go through the process. Is there like a whole panel of comparisons that can be done with mRNA levels, expression levels, protein levels between those cells, the healthy cells, and also normal senescent cells? And I'm not sure. They do refer to a paper they published in the past where they kind of developed this. I think it was from 2016. It's in their references. So I'd have to take a look at that paper if I was so inclined, and see exactly if they, in their methodology, how well they've characterized it. So again, it's kind of hard for me to figure out what's, if this is a good thing or a bad thing. I mean, it seems to be a good thing that you're getting with treatment with THC and CBD. You're getting more collagen production, more hyaluronic acid production in healthy cells, but I'm not really sure what's going on with the senescent cells, these H2O2 cells, in this assay here. And figure 7, it's kind of a bit more the same, but looking at protein levels. So they're looking at, again, the same genes: collagen, elastin, MMP2. Vinculin is, I believe, a cytoskeletal protein that attaches, so it's basically involved with actin filaments. So vinculin is one of these kind of cytoskeletal proteins that is important for a cell to attach to surfaces, right? And if it needs to migrate and move around. And so they've got a lot more Western blots to look at in their supplemental data. One thing, let's see. One thing that I think I'm going to stop sharing here. One thing that I kind of skipped over and I forgot is they mentioned because it's in the supplemental, they actually compare these compounds to rapamycin, metformin, and I believe one other compound, and I think they do a viability assay, and I'm not sure why that's ended up in the supplemental data, because it's kind of an interesting comparison in of itself, because these are compounds that are looked at in the Asian community. Can you see my screen? Am I sharing it now? Yeah. Yep. So these are the images of when they're looking at morphologies. Yeah. So they're looking at wound healing. So this is going back to the wound healing assay that was performed. So one thing that they mention here is that so percent untreated wound, unhealed wound. Let's see, is this okay? So these are population doubling 40. So these are replicative senescent CCD 1135 SK cells. So these are not cells that have been treated with H2O2, but sort of like the more kind of like normal replicative senescence, cells that have undergone many population doublings. And what they mention here is that THC had the most pronounced effect, and that metformin on these cells. CBD didn't seem to have an effect, and kind of neither did, actually, if anything, let's see, vehicle. I believe vehicle will probably be, I think the vehicle is DMSO again. Let's see. I mentioned what the vehicle is.
I do see DMSO down there, yeah.
Oh yeah, DMSO. Yep. Yeah. So let's see, 24 hours percent unhealed wound. So yeah, if this, well, there's a big error bar here, so it's hard to say how much of an effect DMSO is having, but it looks like THC is kind of the most pronounced effect for wound healing, and neither of the other compounds have much of an effect. That being said, I don't know why they didn't look at healthy cells as well, right? Or this, so take this comparison, with the fact that we're looking at these, we're just looking at replicative senescent CCD 1135 SK human dermal fibroblasts, and I'm not sure if that's exactly the same as the other dermal fibroblasts. See, what was the other number that was used for the other ones? It might be just from just a different patient or different sample. Do we have anything in the chat?
No, but Oliver, the error bars are so big that, I mean,
yeah,
this data is like, it's really can't tell us much. The error bars are too big.
Yeah. Well, so that was, they do mention in the paper, and if we can take their asterisks and their, Okay, Mika, take care. Happy New Year. Yeah, so it does leave a lot to be desired there. Let's take a look at, let's go back to the paper itself. Okay, so we have some more issues here with the Western blot. There, I think the authors mentioned that some of the data here doesn't very well correlate with the messenger RNA data. Let's take a look at so this is looking at the protein levels. Trying to see if there's kind of obviously these are supposed to represent differences, but we are trying to compare not H2O2 to untreated, but we're trying to compare it to the THC treatment. So I have, I'm looking at also this Western blot here. I have some questions too as to sort of some of the expression levels and how they're measured. So obviously they're comparing it to the GAP DH, which is the control on the bottom. So for this version of collagen here, A1, you can see that there's a decrease 5, 6, 7, 8, which is H2O2 treatment, and then DMSO. It looks like there's a dramatic decrease. I mean, again, I can't really do this by eye. You would have to quantitate this, and they would probably have to do this with multiple Westerns. So normally how you do it is you measure basically the intensity, and hopefully everything is within the linear kind of zone. Like, you don't want to oversaturate this. So that's the you have to be very cautious when you try to convert data like this to a histogram, right? Because you need to quantitate the amount of pixels that are in an area here. But it looks like compared to, yeah. So if everything in this figure is translated here, I'm just trying to basically look at like COL1A gap DH ratio. So they say here, I mean, it looks like compared to H2O2, which is here, 5, 8 is much more higher, right? If I'm reading this, I don't know if that's really.
Oliver, you have n equals 3 here, so I guess this must be only one of the 3 samples, right? The Western. You don't show here 3, you show here only 1.
Yeah.
It just shows one of them, and
well, you would hope.
Match.
Well, you would hope that it would be representative, right? But yeah. I mean, I guess I have a lot of like other questions here because the authors mention this COL3A1 here, and you do see these prominent 2 bands, and it says 110 kilodaltons, and I don't know what those 2 bands represent. Is one of them an isoform that's phosphorylated, and the other one's not phosphorylated? They don't mention that. Is it 2 different? Is it the same protein but just 2 slightly different isoforms in size in amino acids? I'm not sure what constitutes the size difference. But if you do look at this, you do notice that, I mean, if not to split hairs here, but it is kind of important. The intensity of either of the bands does change, right? I mean, here you have this one band, the top band seems to be very prominent, really expressed, and whatever this bottom band is very light. And then in 5, the intensity of both bands, which is H2O2 treatment, goes down, and then the intensity of both bands shoots up with DMSO. And then for 7 and 8, after THC treatment, the intensity of the bottom band seems higher than the intensity of the top band. So I'm not sure what the significance of that is, and I don't think the authors mentioned that. I don't know if anybody has a comment about that. So anyway, it's hard for me to interpret. is basically it's basically my assessment of this. For vinculin, I'm kind of a little surprised that the levels of vinculin go down. So these are protein levels, or they don't really change too much, because if this is important for the cells to be adhering, to make focal adhesions to plates, you would expect the vinculin levels to go up, because based on their earlier assay with crystal violet, the cells basically had better adherence. So I'm not really sure. It seems to be a bit paradoxical here, right? Why the vinculin levels would go down. Again, not really sure how to interpret that based on their earlier figure. So next, what they look at is effects of phytocannabinoids on mRNA levels of cell cycle regulators. So let me just get my notes here. So they look at mRNA levels, and later they also look at protein levels. Yeah. So let's take a look here. So CDKN2A encodes P16, CDKN1A encodes P21, TP53 encodes P53, right? So, and I believe P53 regulates the expression of P21, I'm not sure if it regulates the expression of P16. But these are tumor suppressor genes, right? So these are involved in basically stopping the cell cycle. NF-kappa B, signaling molecule GDF11, MK16. I believe that's a marker for proliferating cells, if I'm not mistaken.
Yeah, it is.
Yeah. So, again, kind of, if we look at DMSO, there might be a slight effect here, but it looks like I'm assuming that's going to be significant. Again, DMSO has kind of a pronounced effect on the expression of P16. So if we look at healthy cells, you can see THC and CBD increase P16, which seems to be switching on the expression of a potent cell cycle regulator. The same thing with P21. So in general, that's a good thing. In general, I don't know. Again, you have to kind of, yes, tumor suppressors are good, but then you have to ask yourself the question, why is P21 being upregulated? Is it because the THC is somehow doing something negative to the cell and the cell cycle needs to be inhibited? It's hard for me, to interpret. It kind of opens a lot more questions for me. P53 is being activated in healthy cells, right? At least the expression by both CBD and especially THC 2 micromolars. Is that telling you something negative, right? that's happening with these compounds? Should I be worried that P21, P16, and P53 are being just to stop sharing are being switched on? I mean, the cells do that for a reason, right? Generally not for a good reason. I mean, something is happening that's amiss. Is there some sort of mutagenic, capabilities here of these compounds? I don't know. It's hard to say. On face value, you could say, well, yeah, it's great that you're getting tumor suppressors upregulated, but why are they being upregulated? Let's see. For senescent cells, I don't think there's actually much of an effect. At least, yeah, none of these compounds here. For MKI67, so you do have an expression of, so correct me if I'm wrong, Oren. So is this implying that the cells are trying to replicate but they can't because you have this factor being upregulated, but you're also getting P21 and P16 being upregulated as well?
I can tell. I know that when you're doing single cell RNA sequencing, so MKI is like a very well-known marker of cell cycle like proliferation, but actually the P16, P21, P53, I'm not sure that much.
Yeah. So it's well, so they look at the same data using basically proteins, and well, not all the same data. Let's see what they look at here. So yeah, P53, P21, P16, NF-kappa B. They also look at cyclin D1. What do they mention about cyclins? So obviously cyclins are involved in the cell cycle, but there's a particular something in particular they mention about cyclin D1. PCNA is involved in, I think it's part of the clamp complex. It's involved with a polymerase. So I believe a DNA polymerase, right?
Yeah, for sure. It's also one of the main markers for proliferation.
Mm-hmm. So obviously the cells are replicating, so they got to replicate their DNA. BID, what the heck is BID here? Can't remember what BID was. Oh, I'll get back to that later. Let's take a look here. Let's see, cyclin. I'm just trying to. Let's see. What's this figure? Cyclin. We'll edit out the lulls in this conversation in post-production. Duty change to cyclin D1. We just. Yeah, Oren?
They say it's one of the most commonly altered cell cycle regulators in aging and senescence.
Mm-hmm. Yeah, they say that aging and senescence cyclin D1 is overexpressed, but cell proliferation is inhibited downstream, usually by CDK inhibitors. In addition, cyclin DNA acts independently of cyclin-dependent kinases to regulate cell proliferation, growth, and differentiation. Okay, that's interesting. Usually cyclins and CDK, cyclin-dependent kinases, are sort of like chocolate and peanut butter. They both need to form a complex, but it says can act independently, and if it's overexpressed, it's actually proliferation is inhibited because I guess you have CDK, so it's sort of paradoxical, but you have inhibitors that basically block its functionality. All right, so let me go back here. So let's see, is there an effect on cyclin D1 levels? So here, now we're looking at proteins, so different from the messenger RNA. So yeah, clearly if H2O2 treatment, you have a massive increase in cyclin D1, at least on this relative scale to GAPDH compared to untreated. And you do have a decrease that's quite significant, kind of close back to untreated normal cells with these H2O2 treated cells with THC and maybe even CBD. So let's see. I don't know if there doesn't appear to be any really effect on PCNA. Not really, well, H2O2 and CBD. So cyclin-dependent kinase levels go down, CDK2. So does this mean that the cells are replicating less? I'm not sure. I mean, so when a wound healing assay was done earlier, you clearly had an increase in wound healing. So you had more cells kind of fill in with the treatment with THC. So clearly the cells have to replicate to fill in the wound here. So I'm not really sure why you would get why you would get an increase in P21, which would be a cell cycle inhibitor. But when you do the assay for wound healing, you basically get you basically get improved wound healing. So I'm just gonna stop sharing here. It kind of seems a bit discordant. I don't know. Anybody wants to comment about that, right? Because if you're looking at the cells and you're looking at viability assay and you're looking at cells dividing to fill in a space and that is improved, then I would expect to see the markers that are stimulating this, right, that are causing enhanced replication, like PCNA and lower levels of, I guess, P21, like you would expect that to kind of match. But there seems to be a bit of a mismatch there. I'm not sure why.
But here it's not wound healing, right? In this.
No, it's not. It's just looking at expression levels.
So maybe it doesn't have to match it.
Maybe. It's.
But, yeah. And another thing I noticed that PCNA having higher level than GAPDH. I don't know. It sounds a bit weird, isn't it? Also, it doesn't look like it, but.
You mean in the gel image here, or the blot? Or...
Yeah. So in the fold change, they say like it's 1.5. The fold change, which sounds like a lot.
Oh, right here.
Yeah. I don't know. It's like JPDH. I guess it's much more prevalent than maybe not. Maybe PCNA can come higher. I don't know.
Yeah, I don't know what the relative amounts are between the two, but yeah. Yeah, I'm not really. Well, let's see. See if there's anything else interesting here. Well, looks like P16 levels. So here the protein levels kind of skyrocket for P16 in the H2O2 treated cells, DMSO, with THC and CBD. So, I mean, that's probably the most pronounced effect I've seen so far in a lot of these graphs here. But if we go to the messenger RNA levels, what are they? Let's see. I believe that's P16 is which one? K2 or K1? I forgot. It's CDKN2A. So yeah, so if you look at senescent levels full change, there's really no. So there doesn't appear to be any change, right? So I'm looking at this figure here, CDKN2A. So H2O2 THC, H2O2 CBD, assuming that it's 2 micromolar, the same amount. So DMSO treated. It actually, if anything, it goes down a little bit, if that's significant. But if we go to this figure here, we have a several-fold increase, right? P16 to GAPDH, H2O2 THC, H2O2 CBD. And I'm not sure what to make of that. I don't know.
I mean, I think you're too nice, but at some point you have to give up.
Okay. All right. All right. So let's move on. So next they look at sirtuin levels, and these are mRNA levels. Do they look at protein levels of sirtuins? No, they don't. But maybe they should look at protein levels of sirtuins. They look at Sirtuin. So given that there's this kind of discordance between between messenger RNA levels and protein levels, well, let's keep that in mind and take a look at Sirtuin levels, right? So Sirtuins have been in the news for kind of many years. They're certainly Sirtuin is the mammalian version of Sirtu, which was initially studied in yeast. It's an HDAC, histone deacetylase. And they have kind of, they play many roles in aging, in regulation, in gene regulation, stability of chromosomal stability, certainly in yeast. I'm not sure to what extent in mammals. And I know it was studied extensively in David Sinclair's lab, as at least Sirtuin and I believe Sirtu6 for quite some time. The levels in healthy cells of Sirtuin, full change, seem to go up. I'm not sure if this is, we'll take a look at the next panel, if this is translated into the proteins as well. Sirtuin as well. Some of them are localized. I can't remember which one is. Some are found in mitochondria. Some are nuclear. I don't recall all of my Sirt targets here. But the levels, at least, for Sirtuin, at least messenger RNA levels, and Sirtuin as well seem to go up. This is more pronounced in healthy cells. There doesn't seem to be effect in senescent cells, so I'm not sure what to make of that. And if we look at this panel here, which is Sirtuin, there might be a slight increase when we look at protein levels. The one thing that I would say, again, kind of a suggestion to the authors, just because the levels are don't change with the sirtuins or some other enzymes doesn't mean that something isn't happening, because the activity of the sirtuins could be regulated through a variety of compounds. So the HDAC activity, the histone deacetylase activity, could be throttled because it's an NAD-dependent histone deacetylase. So just because the levels of the sirtuins don't change doesn't mean that the activity isn't wildly changing, right? So the HDAC activity might go up, might go down, and the sirtuin levels don't change at all. So I'm not sure if just looking at the messenger RNA level, or protein level of sirtuin will tell you much. Same thing with things like P53, right? I mean, whether it's active or not, the levels might not change, but the fact that P53 is not bound to its inhibitor anymore and is now switching genes on, yes, the levels do go up, but you can certainly, for a lot of these proteins, you'll have effects without the levels fluctuating, and there'll be a massive change in HDAC activity. And there are assays available that you could measure the HDAC levels of the sirtuins by putting in a substrate and seeing, well, is the deacetylase activity changed once I add THC or once I add CBD? So that's something I jotted down as a note for myself. It kind of makes that data a little bit hard to to interpret just by looking at mRNA and protein levels. That's my 2 cents. It's an enzyme. Should look for the enzymatic activity as well, because, like I said, we know that the metabolic state can definitely influence sirtuin activity, and other compounds can affect sirtuin activity. So maybe THC and CBD are doing that without altering the expression levels significantly. Last, what they look at here.
You're not sharing.
Oh, sorry. I don't know why today I'm forgetting to share. Maybe the button has changed or something, or maybe I'm just. My apologies. Okay. So last, what they look at is the effect of these phytocannabinoids on cannabinoid receptors, right? And this is also where we're looking at the sirtuin levels as well. So I mentioned that there's CB1 and CB2 receptors. I think CB1 is can't recall if THC binds to both of them or CBD binds primarily to one or the other. I think maybe CBD binds to CB2. I think they're both G protein coupled receptors, and this is how they interact, through THC. Now, what the authors are doing here is a Western blot. So they're looking at, they're saying that the levels are not really changing. I'm not sure if that's the best way to look at. I'm not sure based on past papers and studies whether or not the levels of a receptor would change dramatically if you added THC or CBD. Maybe a different assay like immunohistochemistry to look at actual receptor expression on the surface of the cell would be a better way to visualize this, right, than just total protein levels of receptor, because you certainly can have receptors recycle underneath the surface of the cell and be sequestered, depending on whether or not there's more or less of something binding. So again, kind of looking at things a little bit more subtly, if the levels of a receptor in total levels of protein don't change, it doesn't mean that there's the same amount of receptor on the cell surface, is what I'm saying. There could be much less or much more on the plasma membrane, based on whether or not the receptor has been kind of internalized. So that's kind of my 2 cents here. So yeah, what else do they look at here? And they look at also epidermal growth factor receptor levels. So again, surprisingly, the protein levels seem to go down, I think, with THC, and I'm not sure why. If the cells are proliferating more, I think I would expect them to be a little bit more responsive to epidermal growth factors, so they would upregulate this receptor. I think the bottom line is a lot more needs to be done with CBDs and THC compounds, and to see what their effects are on senescent cells. I think what was clear from this paper is that the cells replicated more in a wound healing assay. So clearly the THC and CBD are having some effect in that sort of tissue culture based assay for replicating cells, and that their viability is increased. What is underlying that? I think it's a bit hard to interpret. We do see an effect from DMSO, the carrier. I think more kind of control should be done to see if a better carrier could be used, or why this effect is happening with DMSO. And it's still not clear to me what effect THC and CBD really has on senescent cells, and if that effect is a good thing, right? If the CBD and THC are protecting senescent cells, do we want those cells to be protected? Or are they improving the senescent cells so they're becoming less senescent? So I think looking at kind of a whole palette of senescence-associated secretory phenotype factors and seeing if the profile changes with these senescent cells, I think a lot more needs to be done experimentally to really get down to the bottom of if these compounds are actually having an effect on senescent cells. Is it a beneficial effect to an animal that has these senescent cells? And I don't think I think it's very unclear from the data here what the answer to that is. But I would like to see a follow-up paper where they really kind of drill down deeper to figure out if these effects are real and whether or not they're truly beneficial. So it's still kind of up in the air for me whether or not these effects that we've seen are replicatable in other cell types as well, because again, they only use this one cell type and they use this H2O2 assay. So I would love to see actual senescent cells, maybe from even cells from older individuals, as well as cells that have gone through multiple population doublings from different tissue types. Look at a much wider expression profile. So kind of look at every mRNA, because we can do that now. Look at the expression of every mRNA, right? And see what the effects are, rather than looking at just a few genes, and see whether or not you can compare, like, apples to apples, whether or not you have a reversion of these senescent cells back to a non-senescent expression profile and a non-senescent phenotype. And it's not clear to me right now that this is indeed what is happening, because that would be the beneficial effect you would hope for by taking THC and CBD. You certainly wouldn't want to be increasing your load of senescent cells, right? So those are my comments. And I'm open for discussion right now, or not.
I mean, it's like what you said. It was interesting that the part that they actually had the phenotypic effect, but except for this phenotypic effect on wound healing, so it seems like we needed some more phenotypes here, and it's not clear what exactly we're looking for.
Yeah.
Yeah. So.
Inconclusive. Is how I can put it, and I would really like to see a follow up work with cannabinoids, because people, they're in so many products. There's so many health benefits being touted for these different cannabinoids, and it's practically everything, right? I just was joking at some point that I think even my car wax has cannabinoids in it, right? So CBD infused car protectant. So I mean, so is it, so what is exactly going on, and can we, is there any other than increased wound healing in this one assay? And I'd like to see the wound healing assay. I'd like to see repeated in an animal model, for example, right? So you can certainly do with mouse models to see if you have increased ability for wounds to heal in the actual, dermis and epidermis of an organism, rather than a scratch test that you have on a plate. So yeah, I think just for that reason alone, that these compounds are so prevalent now in society and are used so extensively, and the concentrations people take are wildly kind of varying in range, right? When people take CBD extracts, I think really there's a great need for more work like this to be done, that could really get to the bottom of effects on aging and senescence. So yeah, unfortunately, the data here was inconclusive to that end. All right, that's all I got.
So, it was a short paper, right, Oliver? 3 hours later.
I wouldn't say it's a short paper. Yeah.
What was all this talk about? It'll probably be quite a quick one today. Yeah.
Yeah, I always say that, but
I know.
Yeah.
We know, Oliver. There's no such thing as short when science is on the line, right?
Yeah. So, yeah, as was mentioned before, it's a good thing that the authors showed the DMSO control, because DMSO is having an effect, and it'd be interesting to see the senescence profile of cells treated with DMSO. I don't know. maybe DMSO has anti-senescence properties. I don't know.
Well, that also brings on even more worrying questions, as DMSO is used quite a lot in experiments in general, especially mice for topical applications. So yeah, that's a worry.
Yeah, maybe. I mean, yeah.
Well, if it's having an effect, a significant effect, it certainly is a confounding factor, isn't it? So
yeah,
that is a problem, because I know that some senolytic studies use DMSO.
Yeah, well, as long as they showed the control with the carrier alone, then and there wasn't too much of an effect, then I suppose it might be okay. Maybe, I don't know. Maybe those two compounds together have some sort of unknown combinatorial effect that we're not aware of.
Yes, it could be additive or synergistic as well. So that is again another potentially problematic issue. So there you go.
Yeah.
So there you go. The conclusion is there is no conclusion. Dare I say, more research needed.
Yeah.
Yeah, more research needed, of course. Come on, guys. We all knew this is the punchline.
Yeah, so unfortunately, this wasn't the THC and CBD paper I was looking for, but hopefully the authors will do a follow-up where they can address those concerns, and I'm optimistic that in 2023 there will be more data on CBD and THC that can kind of pin down its effects on senescent cells and aging in general. Kind of little move the needle a little bit more clearly in the yes or no direction. So that's my prediction for the new year.
Well, it worked for the Rolling Stones, didn't it? Because I'm willing to bet they smoke plenty of cannabis. So, we could do a field study on Mick Jagger, yeah?
Sure. Yeah.
He's really 150. Yeah. But actually, joking aside, Mick Jagger is known to have tried some anti-aging things. He was well known. I can't remember what it was in the 70s or 80s. He had a form of blood exchange, a bit like a la Comboys, but an earlier version of it.
Really?
Yeah, I think he went to France and he had some a blood exchange or something like that.
How long ago was that?
I think that might have been in the 80s. I'll have to look into it.
That's a little scary, considering that they probably had very poor HIV screening back in those days. Yeah.
And I understand it was somewhere in France, but I also understand it may have also been like a young person's blood. So yeah, but now we know what we know about neutral blood exchanges. It was obviously clearly closer to the mark than some might have thought. But yeah, don't rush off to France and get a blood exchange.
There was a famous scientist who did so. These blood exchange experiments go back quite a ways, and I don't think it was Eli Metchnikoff. It was probably somebody else, but it was somebody. It was kind of it was going back a ways that this scientist did a blood exchange, and anecdotally, he said it felt he felt improved. I have to find this story, but he unfortunately did it later again, and he died because the patient that he received the blood from had malaria, and
oh boy,
and he caught malaria. But I have to. So whenever I hear of these blood exchange stories, I always think you really gotta screen where the source is coming from if you're going to do this type of experiment.
Absolutely, and on that lovely thought. Well, I guess this is the last one of the year, isn't it?
Yeah.
So we will return
later in January, way at the end of January. So everybody's going to have a nice long break. Wishing everybody Merry Christmas, Happy Hanukkah, Happy Kwanzaa, any other holidays that you may or may not be celebrating or just taking time off. Maybe you're going to hit the ski slopes. But anyway, I wish everybody a happy new year. Hope
and a healthy one.
And a healthy one in 2023.
And a healthy one. And everybody out there, make sure you do take the opportunity to rest and relax, which is very important for longevity. Even we're going to be taking a little break. We're going to be taking this Friday. We finish up and we're back on the 3rd. So don't panic. There will be some things coming out on the website as well for those of you who absolutely can't live without the news for a week. We've got some stuff. We've got you covered during the week. We will see you end of January, probably with something exciting, I would say, paper-wise, because there's always plenty going on. And so stay happy, stay safe, and stay healthy. And we will see you all in the new year. And thanks to the Lifespan Heroes who support programs like this and all the work that we do. And if you're interested in learning more, you can find more out at lifespan.io/heroes. And hero as well. They both work. I know they did because I made them both work a couple of weeks ago. We will see you all later.